then the chemistry part here in the middle, and then the pharmaceutical sciences, safety and toxicology part, and here would come then the entry into human and the clinical development over there outside into the garden. So, first of all, we need to understand a disease by its mechanism. So we want to understand the disease on a molecular basis. And here you have a nice picture of a cell, for instance, and we want to identify potential points of intervention to as a therapeutic modality, and it can be an intracellular point. This is a target then, an enzyme, for instance, or a cell surface target, can be a channel or a receptor, or it may be even an extracellular target, something that swims outside a given cell. And the target may be hit by a small molecule that penetrates into the cell and blocks here, or it may be a biomacromolecule, for instance an antibody, and that would block here the receptor and block then the signaling. Now, you see, this is a very complex construct, this cell, and you can imagine that all proteins in here, all these various things, interact with each other, and there are very complex processes going on with many feed forward and feedback loops, positive and negative loops. It's a very complex system. And therefore, nowadays, what one engages in is what is called systems biology, a very demanding and very challenging activity to understand what's going on in living cells, in our tissues, in living organisms. And by all this science, cutting-edge science, not only in industry, but also, of course, in academia all over the world, one tries to understand if what goes wrong and what could be the molecular basis for a disease, identifies maybe interesting intervention points, the so-called targets, and then starts later on, if the target assessment is positive and done, so that we confirm that this is a valid target to maybe block, then of course we have to develop assays, and we do perhaps screening, and come later on into chemistry to produce small molecules to block that target. Or we go into biotechnology and try to generate antibodies or other biomacromolecules to block other targets. Now, of course, there may be a bioactive natural compound, whatever the source, from microbial sources, from deep sea organisms, or from plants, or from animals. And typically, such bioactive compounds may be cytotoxic, and then very quickly academic groups say potential anti-cancer drug, and this sets then the idea in the mind that, you know, if something is there, it is already virtually a drug. This is not true. Typically, these are cytotoxic. Many of these compounds are cytotoxic. But they may, we do not know the mechanism of such bioactive natural compounds typically in the first place. And it needs a lot of this kind of research to find out where this molecule, this natural compound, actually interacts and interferes. So this research is typically not done at Roche, I must say. Maybe in other companies it is done, but we leave this to academia. And it takes maybe 10 years, maybe 15 years, sometimes a little less, to find out the mechanism of a natural compound and its bioactivity. If that is identified, it might provide new ideas, new intervention points, new targets, and then what we discussed for here may then be a starting point here to try to do with chemistry, find new molecules that have a bioactivity. Now, what is involved? All biology, biochemistry, bioanalytics disciplines, I will not go into these things, but also genomics, proteomics, genetics, more modern type of developments, high-tech developments. Pharmacogenetics, very important to differentiate between different groups of patients. And very recently, there is this fantastic new sequencing methodology, 454 life science technology, where now we can do whole genome sequences, the whole genome in a couple of months. And I'm very positive in a short shortly we will see that one can do a whole genome perhaps within a week or weeks. Then, of course, biotechnology, you have to be able to express and produce the target for your studies. We do protein X-ray structure, structural biology to see the three-dimensional structure. This is now very, very important, and also for the screening here. Finally, biomarkers research, the whole expression profile in an organism or in cells, in tissues, and we do that not only in tissues and cells in industry, but of course also at the clinical settings. So this is exploratory clinical research to make sure that what we observe here, say in given cells or tissues or in animals, can also be found and correlated in actual patients that are analyzed. And this is now a very important area of translational medicine, where you try to couple the clinical research with the early discovery research. And there's more than just gene expression profiles. There's imaging by magnetic resonance, by confocal laser scanning microscopy, analytics by mass spectrometry, very high-tech, and there are many, many more approaches to make sure that we have a direct link between the clinical setting and say the human picture and the early cell, tissue, and animal picture, to make sure that what we focus on here on this target has a correlate later on in the patient. So all this is underpinned by now a very heavy activity in so-called bioinformatics by computer, where now, because we have many genomes now available, comparative genomics, where you identify more things than before, particularly non-coding areas can be analyzed. We can go into gene regulatory mechanisms, and it's also very important for this biomarkers research. So this is the whole bio picture, including this one. Now, coming to the second stage, chemistry. Chemistry, of course, we have now typically in 50% of all our projects, we have the three-dimensional structure of the protein available through X-ray structure analysis. It's a heavy investment, but it can be done. And then you have the three-dimensionality of your protein, your target. You see the pockets where things bind, and you can use directly computer-assisted molecular modeling in this pocket to design an optimal ligand. And methods have been very nicely, of course, refined in that area. If you do not have that information, you still know a lot about the biochemistry of your target, and you know about natural ligands and natural substrates of an enzyme and so on, and can use that information to start your design. So target biochemistry-based design. The opposite is an ultra-high throughput screening of large compound libraries. I talk about million compounds, which you may simply screen without any preconceived notion and see what might be active in this compound library. And we will come back to that, what is in this compound library. But we found that actually more convincing than just doing this blindfolded massive screening, which is quite expensive still, is a ligand motif-based molecular design. Proteins come in families, and if you know one member of that family, you can deduce that other members of that family, which may be of interest to you, will have the same motifs, structural motifs in your ligand, and you may actually do a motif-based molecular design and generate molecules that have a high chance to interfere and bind to that specific target. And in the same vein, we do not the big library, but focus libraries, where we concentrate on interesting motifs, or what we call privileged structures. These are structures that every so often turn up as interesting compounds interfering with proteins, and we can generate libraries beforehand, small libraries, say 50 to 100 compounds, that are centered around given privileged structures. These are all ideas, and they're not natural compounds. They're just de novo designed and synthesized to make sure that we have a higher chance by screening to find interesting active compounds. You can do the whole thing nowadays with computer alone, just virtual libraries. That these compounds are then not existing. Millions, multi-millions of compounds by computer, in the computer, and then use chemoinformatics to weed out the interesting ones and do screening by computer, within the computer, no synthesis whatsoever. And from the results here, then you will start a focused synthesis of interesting members. Finally, I should not await this one, natural compound focused libraries. So you can take a natural compound and try to either synthesize it and synthesize congeners, or take the compound itself and start to do chemistry all around and try to modify and see whether among these there are interesting bioactive compounds. Now this is largely done in academia, and the question is, do we do it? I can give you the answer, no. And why? See, we are very successful now in this area, we are very successful in this area, we are very successful in this area, we are very successful with this one, moderately successful in the ultra-high throughput screening that we only do if you really have problems. But here with natural compounds, it's a lot of effort, it's cost and time compared to these, and typically natural compounds by evolution were not done to cure a disease of man, but they are produced in nature for other reasons, so typically they do not have proper pharmacological properties. And in order to find the right properties, you have to modify this compound, and maybe you lose potency. So this is a lot of effort, and typically has been in our hands 15 years back and more, not very successful in comparison to the other one. These have really improved, and these are now the front runner in terms of success, so we are not doing this one. To give you a perspective, ultra-high throughput screening, I mentioned this million type of compounds. Right now we have something like 1.2 million compounds in our compound repository, and focus libraries are typically in the order of 100 or less compounds, but these are really high quality compounds, purely synthesized. And you see that this is reflected in the hit rates. The hit rate here is considerably less than 0.1%, whereas here typically it's 5 to 20%. If you have hits here, you have much better starting points for your research. When you look at the compound library, these 1.2 million compounds, you see these are internally synthesized, these are by contract synthesis with external groups, and these are bought-in compounds, all de novo synthesized. And what you would call natural compounds, this is a slice of 0.2% or less, so virtually nothing. And when you now see this hit rate here, then you can easily calculate that the chances that you ever buy full-blown ultra-high throughput screening of this current library, that you would hit a natural compound is 1 in a million. So it virtually never happens. And even if it happens, then of course you have other hits which are not natural compounds, and typically the non-natural compounds, the de novo designed ones, they win over the natural ones, because it's so much easier to actually improve and work chemically in a short time line on these and be successful. So, of course, you need a lot of technologies, analytical technologies, and you need protein ligand interaction analysis and so on. I will not comment on this. Rather, I want to focus. The most potent compound is not a drug yet.